tell us a little about your work.
I study neuron degeneration, and specifically it's about formation, regulation, and function of axon spheres in the process of axon degeneration. In reality, axon spheres is not a new idea. It has been identified by Cahal like a century ago. But the thing is, the regulatory mechanism and the function of them in degeneration remains unclear. So my dissertation project is sort of answer those two questions. So the thing about axon spheres in degeneration is it actually a universal hallmark across different species in different type of neurons in CNS and PNS. So I use specifically sympathetic neurons as a model to understand that, to figure out that process.
So did you do experimental work with the mice? Was this mostly observational?
Yeah, it is. I would say 90% experimental. So I would dissect the superior cervical ganglion neurons from P0 to P2 mouse pups and culture them in a dish, and then do different manipulation on them. Most of my work was based on live imaging data. So I use a lot of microscopy technique.
How did you get interested in this topic, and how did you find the Deppmann lab?
Yeah, back to my middle and high school date, I'm intrigued by a painting of neurons in brain slides. It's nothing scientific, but it's just they paint the neurons as a star in the sky. It's just beautiful. And then I get interested in neuroscience and actually record the neurons and seeing the firing pulses by microscope and my own eyes, not from textbook. And that's actually a real excitement, and it's a feeling of fulfillment. I looked into his website and get intrigued by his papers and the groundwork. So yeah, that's how I joined Deppmann Lab.
And you're just finishing up now.
Yeah, I actually finished my defense late October. And I'm heading to San Francisco to start my postdoc work soon. It's gonna move towards central nervous system. It's about the brain, and it's more gonna focus on immunology, so brain immunology, what's happening on the glia side, microglia specifically. So I think I'm gonna study the communication between neuron and microglia in neuron degeneration and disease model like multiple sclerosis.
What are you gonna miss most about the Deppmann lab and being in Charlottesville?
Firstly, the people, my colleague in the Deppmann lab, they're super nice. And it's my pleasure to work with them. The core values in the Deppmann is like open-minded, always be friendly, always ready to help each other. And I really appreciate this opportunity to be part of the Deppmann family. For Charlottesville, I really love the fall and spring in Charlottesville. It's a cozy small town, and it's got everything you need for living and working as a graduate student in this town. So yeah, I'll miss here.
And any other advice you'd have for anyone considering grad school in the Deppmann lab or at UVA?
It depends on where you're at. If you are trying to apply in graduate school, I would say UVA and biology department is a very good place to join. Talk to your other graduate students in the department and ask their opinions. And also talking to professors even before you join the lab. And thirdly, I would say take full advantage of the rotation process. I know you're trying to impress the lab and the professor, but the lab is also trying to impress you. So take that.